Thank you for standing by, and welcome to the IMI webinar on Regulatory Acceptance Mechanisms conference call. At this time, all participants are in a listen-only mode. There will be a presentation followed by a question and answer session, at which time, if you wish to ask a question, you will need to press *1 on your telephone. I must advise you that the conference is being recorded today, on Thursday, the 23rd of May, 2013. I would now like to turn the conference over to your speaker today, Magda Flebluff. Please go ahead.
Thank you very much, and welcome to everyone to this webinar, which is co-organized by three organizations, EFPIA, IMI, EMA and FDA, with the objective of discussing the regulatory acceptance mechanisms. The reason for that, and you would see it on the next slide, which is showing up, is that in IMI, the way we measure success is through practical application and implementation of research results that come out of IMI projects. And as you can see on this graph, this is not just about developing a new model or a new standard or in-house implementation by the industry, but also this becoming a wider practice in the regulatory setup, as well as having a concrete impact on clinical practice. And this is one of the main reasons for us to have this webinar today with you. At this slide, you see the program of today, which would be basically in three parts. The first part would be an explanation by Nathalie Seigneuret from IMI about the types of tools, information, mechanisms that you, when you develop it, you should be aware that they may be interesting for the regulators and therefore could run through an informal or formal dialogue with them in order to get a regulatory acceptance for those. Then we would go through two sessions, one on guidance and one on a more formal qualification programs in both EMA and FDA. Each of those would be followed by a Q&A session, and the procedure for asking questions would be reminded to you after each of these sessions. And finally, we would come to a brief summary and information or where to find more information and on follow-up actions. We've prepared a couple of pictures for you to know who is talking to you and to put names on faces and faces on names. So on the first slide, you see myself, Magda Flebluff, Director Science Policy from EFPIA. One of the next speakers would be Nathalie Seigneuret, the Scientific Officer at IMIGU. Maria Isaac, a Scientific Administrator at EMA, as well as Torsten Vetter. Unfortunately, we don't have his picture, but he would be also presenting on behalf of EMA. And finally, from the FDA, we would have two presenters, Amita Parikh and Shashi Amor. So you know how we look like, you know what the program is, and I would now hand over to Nathalie Seigneuret, who would introduce the first presentation. Nathalie, over to you.
Thank you, Magda. So this first presentation is much more an overview of the IMI project, and as well to give you a sense on why it is important to work with the prospect of going to regulatory acceptance of the output of the IMI project. And, sorry. Okay. And as you may know, because most of you now are part of the project, we are currently 40 ongoing projects from call 1 to call 6. The one coming out from the call 7 are not yet started. Out of this 40 ongoing project, you may see that it mobilized a number of teams coming from academia, from industry, SMEs, but as well regulators and patient organizations. So all in all, we have more than 4,000 researchers involved in the IMI, which is quite substantial. And of course, this project aims to improve the research and development productivity of pharma industry, but to find out as well innovative approaches for developing medicines for unmet public health needs. This slide just to give you a brief overview of the different projects. Of course, here you have to note that the one coming out from Call 5, which is the European Lead Factory, and Call 6 from the antimicrobial resistance are not yet plugged in here. But this gives you an idea that most of the projects you are involved in are tackling a different aspect of the drug development, being from identifying predictive pharmacology in vitro, in vivo, biomarkers discovery, clinical development, chemical and pharmaceutical development, risk assessment, and some of the projects are actually addressing various aspects of this line chain. So it is important to keep in mind that these projects are covering several important aspects on the drug development, and there are measurable outputs which are coming out, especially from this project of the Call 1, but started as well from the Call 2, and of course it will become more and more as the projects are going into their duration and their life. So some of the measurable outputs of the project are hopefully will be validated robust models for drug development, biomarkers and tools predictive of clinical outcomes, both efficacy and safety, potential new drug targets, improving the design and process of clinical trials, the big data solution to leverage knowledge, and education of training for new generation of R&D scientists. From out of this measurable output, there are some which are of regulatory relevance, and this is important for you in your project to identify what would be of regulatory relevance. And of course, we will discuss the qualification program of biomarkers. Most of your projects are addressing the development of new biomarkers, but as well new models, either in vitro, animal models, in silico models, clinical outcome assessment, and we have some projects developing patient-reported outcome. Modeling and simulation is an aspect as well addressed by most of you in the project, and tools and methodologies, whether imaging, diagnostic, assays. Some of your projects are addressing actually several aspects of this measurable outputs of regulatory relevance, so it's not necessarily only one, it can be more than one. We have as well projects looking at new approaches for clinical trials, and I just listed here some of the projects: autism, schizophrenia, antimicrobial resistance, to find out new ways to improve the design and the process of clinical trials in this area, but as well projects addressing standards and consensus definitions. Like we have definition on severe asthma, which may have an impact as well on the way of conducting the trials. So as Magda already presented, this is an important aspect to realize that there will be a need to translate all this research into the regulatory and the clinical practice. So it's important to engage as early as possible with the regulators for those projects which have or may have outputs of regulatory relevance, because it's important that this output will be actually accepted. Why? Because it gives the assurance of the scientific rigor under which they were developed. It gives the confidence of their value and that they can be used to increase the certainty and the predictability, that this tool which will be developed will be translated into practical application and not just remain as a nice publication. And what is hoped is that they become the standard for the whole scientific community to be used. But it helps as well by engaging with the regulators in their role of supporting innovation and giving the assurance of their understanding and acceptance of their validity. And on the whole, it's to give an overall impact on the overall regulatory requirements. So these are important aspects to consider, and I will just finish by saying that the scientific progress and the regulatory science have to work together, as it will be the way to accelerate the pace and reduce the cost of R&D to bring innovative medicines for the benefit of patients. And at this note, I will now move to the next session to give you some idea on how you can engage as early as possible with the regulators for advice and guidance. And I give you the floor to Henry and to DMA.
Thank you very much, Natalie. And I think while the floor is given to two EMEA colleagues, I would like to just introduce the next presentation on qualification advice on protocols and methods for the development of new biomarkers and methods in drug development, which will be given by Torsten Vetter from EMEA. So Torsten, the floor is yours.
Thank you very much. Yeah, so apologies for not presenting in person on a photograph, which I didn't have at hand in time. My name is Torsten Vetter. I have a background in clinical and experimental pharmacology and as well in internal medicine and endocrinology. And today we are very happy to present to you the EMA qualification process on protocols and methods for the development of new biomarkers and methods in drug development. This is a voluntary scientific pathway which leads either to a qualification opinion or a qualification advice on any kind of drug development tools. We have split the presentation according to the schedule of this webinar and between ourselves, between my colleague Maria Isaac and myself. I will rather introduce you to the principal structure of the European regulatory environment in terms of scientific advice and qualification advice, and try to introduce you to the procedural aspect of qualification advice, while Maria will take care of more aspects on the qualification opinion later. So let's go to the next slide. So what is the EMA, the European Medicines Agency? It's an agency which is acting as an interface of cooperation and coordination of the member states' national competent authorities of, as of now, 27 member states with respect to medicinal products. It makes use of the European Expert Network, and this network underpins the work of the agency's scientific committees, of which we have 7 currently, 6 of them concerned with human medicines and 1, the CHMP, with veterinary medicines. Overall, we have listed roughly 5,000 nominated experts, which again support the agency in any kind of scientific assessment. To provide you with a short overview of the 6 committees concerned with human medicines that I mentioned before, we first have probably the most important committee, the CHMP, the Committee for Human Medicinal Products, which in the end is in charge of assessing any kind of marketing authorization application and granting authorization within the European Union. Then just briefly to mention, we have the COMP, the so-called COMP, which is the Committee for Orphan Medicinal Products. We have the HMPC, the Committee for Herbal Medicinal Products, the PDCO as a Pediatric Committee, the so-called CAT as a Committee for Advanced Therapy Medicinal Products, and just recently come on the stage the PRAC, the Pharmacovigilance Risk Assessment Committee. When you look in the upper left corner of this slide, you see a little satellite and an acronym SAWP, so you surely, most of you hopefully will know this acronym. This is for Scientific Advice Working Party. The Scientific Advice Working Party is a standing working party of the CHMP established by the Regulation 726/2004, and is the general access point to receive early scientific feedback on all aspects of drug development. And the Scientific Advice Working Party basically connects and combines all the different groups and working parties within the European regulatory system to provide you with the most valuable early input into your development programs. The Scientific Advice Working Party currently consists of 28 members. It's a multidisciplinary expert group, and the membership is not depending on member state status, but strictly on expertise. And this is important for you to keep in mind. You have scientifically very knowledgeable and very experienced people as members of this group who will scrutinize all documentation received according to their very high scientific standards. So currently we have 16 members that are staff from national competent authorities, 12 members who are academics, and then we have 3 members from the Orphan Committee, 1 member from the Advanced Therapy Committee, and 2 members from the PDCO. And of course, the Scientific Advice Working Party makes use of the extensive European expert network. I think, as I said, the Scientific Advice Working Party can deal with any aspect of drug development. I think I don't have to go into the examples of areas for advice. I just wanted to mention probably some obvious and frequently discussed topics like for quality, starting materials, specifications, comparability, bridging, in terms of non-clinical development, the overall toxicology plan, registration, innovative models, in terms of clinical pharmacology, PK/PD development, modeling and simulation, bioequivalence questions, for clinical therapeutic areas, of course, the questions for adequate endpoints in pivotal trials, population, comparator, and of course, paramount questions on methodology and statistics concerning, for example, interim analysis, adaptive and seamless designs. It is important to remember that the Scientific Advice Working Party is very closely linked to the CHMP. The CHMP role is to peer review all the reports that the Working Party produces and to discuss before adoption the final advice letter. This is just a diagram to show you that we are very happy to see that this instrument of scientific advice and protocol assistance being scientific advice for orphan drugs as a specific procedure, is taken on very well from the industry side. And then you can see how numbers have increased. We are roughly between 400 and 450 procedures per year right now. This slide tries to introduce you to the number of specific, specialized scientific groups that are orchestrated within the EMA network and that are accessed via CHMP and Scientific Advice Working Party. The CHMP is, as I said before, the central decision-making group that takes advantage of all these different working parties and scientific advisory groups, which I will not mention in detail here. Probably just in the context of biomarker development and novel methodologies, I have highlighted groups that are very important in our qualification process. And starting from the left, this is the Scientific Advisory Group on Diagnostics, the newly introduced working group on modeling and simulation, the pharmacogenomics working party, and of course the biostatistics and methodology working party. The need for a dedicated process to channel feedback for the development of novel methodologies was recognized years ago, and after internal discussions on how to best implement it led to the development of and introduction of the qualification procedure on novel methodologies, which started formally in 2009. This procedure is basically using the framework of the Scientific Advice Working Party and is following the structure of the scientific advice procedure as far as possible. But I will be happy to try to outline the differences. So let's have a look at the table on the left side of the table first, which is depicting how scientific advice and protocol assistance procedures work. So scientific advice and protocol assistance is product and indication specific. The procedure has 6 timelines, 40 or 70 days, depending on whether the working party considers a discussion meeting necessary with the applicant. The applicant can intervene only if requested by the scientific advice working party, mainly in case of disagreement with the proposal. The scientific advice working party looks into the data but focuses on the methodology and formally by mandate cannot do a formal pre-assessment of the data submitted. Scientific advice and protocol assistance is always confidential until the drug is approved. Now let's have a look on the right-hand side of the table where the differences for the qualification, advice, and opinion procedure are described. So in terms of scope, the scope of the qualification, advice, and opinion is broad. It may concern several indications, several products, a class of product, novel methodologies for specific R&D requirements, safety signal detection tools, and, as I said, any kind of drug development tools. The timelines, I will present you with some timelines which will be ideal timelines in the sense that these are the shortest timelines we can possibly finalize the process. But it's important, and we would like to stress that these timelines are absolutely flexible and the proceedings are flexible and will be tailored to the needs of the procedure and to the needs of every single application. Within the qualification advice procedure, we always have face-to-face meetings in which the applicant can raise issues for discussion and add additional data that may have become available meanwhile. And importantly, of course, and that's why it is depicted in bold here, the qualification, especially the qualification opinion procedure, is always an assessment of data. With regards to the outcome product of the process, in terms of if CHMP, scientific advice working party, at the end of the process decide that the new drug development tool is sufficiently mature and robust and underpinned by sufficient adequate pivotal data, this ends in a positive opinion which will be publicized. However, at the beginning of the procedure, we do not need to decide on which way the procedure will go. So basically, if it turns out that the data may be viewed as not entirely sufficient at the time, we will always be able to change the procedure to a qualification advice procedure, which outcome as the qualification advice letter will be confidential and not published. So I think I don't have to go in details here as well for such a knowledgeable audience. Just some examples of qualification for topics for biomarkers. For the preclinical development, we have seen pharmacological screening tools to better address and investigate mechanism of action, predictors of activity and safety of drugs, PK/PD modeling, toxicogenomics are usual topics. In terms of clinical development, very important, models biomarkers to verify the mechanism of action, to facilitate the investigation of dose-response relationships, to facilitate the proof of concept studies, tools to enrich population, to specify population, the development of surrogate endpoints, and tools for the early detection of safety signals. Later on, markers or tools may be developed for drug utilization improvement, as for example, to optimize target population for the application of drugs, and tools to guide treatment regimens. So here, so as I alluded to before, for the European Medicines Agency Qualification of Novel Methodologies procedure, there are two possible outcomes. There can be a qualification opinion on the acceptability of a specific use of the proposed method in R&D based on the assessment of data and not product-specific, or there can be a CHMP qualification advice on future protocols and methods for further method development. And as I said, if an applicant started out requesting a qualification opinion and it turns out that there are some more hurdles seen by the regulatory system, we are always in the position to change the procedural target towards the qualification advice. The aims are that, as Nathalie has already also pointed out, there is a huge interest on our side to have an early involvement in the design of the strategy, and from our side this will then create the commitment to evaluate data generated in your development approach from agreed studies and to provide a positive, hopefully positive qualification opinion. So who is doing the work? I just jump 2 slides now and go 1 back because I think it's probably easier to understand this way. So we have the core group dealing with any kind of qualification procedure is the qualification team. It is comprised of 1 coordinator, several team members, who are selected according to expertise and according to the procedural requirements, and 1 EMA scientific administrator. And the role of ourselves, the EMA scientific administrators, is of course very important because we are the primary contact person for any applicant, and we are very happy to provide scientific, administrative, and logistic support at any stage. As I said before, this whole procedure is embedded in the scientific advice working party, which serves as the primary scientific group, and which allows extensive networking within the agency, as I have shown to you before. The CHMP is primarily involved. The CHMP member, we always have 1 member of the CHMP as a team member. We have the CHMP peer review. We have CHMP discussion and adoption of the final responses. And of course, coming from the qualification advice procedure, a thorough qualification advice will always lead to the commitment of SAWP and CHMP to continue the interaction and to evaluate the data obtained from studies which have been agreed during the qualification advice procedure and to provide a qualification opinion later on. Just probably, again, to clarify the structure of the qualification team, I think that is quite important practically for any one of you planning on coming for a qualification advice. So we have the coordinator, who is a SAWP or CHMP member. We have the product manager from the EMA providing scientific, logistic, administrative support, and we have at least 4 other experts defined by the therapeutic area concerned. We have 1 statistician methodologist, and as needed, really experts on the technology platform concerned, and if there's any special needs defined by the context of the intended use of the tool. This slide, and I'm sorry that it's, at least on my monitor, it's looking quite small. Probably if you can have a printout later, or you can have a look in the published guidance where this is as well. It shows you roughly the timelines that we foresee for these qualification advice procedure. So it would be good if you contacted us roughly 2 months before you plan to start your qualification advice procedure. This can be with an informal email, and we usually will respond very quickly in clarifying, potentially in a personal conversation, how to proceed further. If you have any doubts whether the stage you are in is adequate to come for a qualification procedure. Then if you decide to come for a procedure, we ask you to submit a letter of intent and a draft briefing package, which will then be reviewed, and you will receive validation responses. And usually in the last month before we start the procedure, we will have one meeting, which can be in person or as a teleconference. We are very flexible there, to provide you with feedback on the package in order to address potential gaps or to improve the content of the package. Then at what we call day 0 here, the procedure starts, and then our experts, the qualification team, has 1 month to assess the package and come up with initial comments, and in the Scientific Advice Working Party meeting 1 month later, we'll discuss these comments and come up with a list of questions to the applicant. We will invite the applicant, again 1 month later, this is then day 60, to a face-to-face meeting in which we will be happy to deepen the insight and to address the issues identified earlier by the qualification team. If everything goes well, I will now, the qualification team will come up within the next 30 days with a draft report, which is discussed by the Scientific Advice Working Party. And if we look at qualification advice, we'll then go to the CHMP for adoption. I leave out the qualification opinion timelines here. This will be addressed by Maria. As I said, the timelines are very flexible. Documentation needed is letter of intent, briefing document, supportive documents, scientific literature. Applicants can be consortia, networks, public-private partnerships, learned societies, academia and pharmaceutical industry. The fees are basically the scientific advice fees with all the fee reductions for pediatric, orphan conditions, and SMEs as usual. Probably I can skip this. Importantly, I think that is important to mention, we have the possibility, and this is frequently used to have the qualification advice procedure as a parallel FDA-EMA qualification advice. This is a voluntary, of course. The applicant will have to request this from both agencies independently. Questions on the this will lead to involvement of FDA at the time of the discussion of the first assessment, and we will have the face-to-face meeting as a tripartite meeting. This will, of course, increase the dialogue between the agencies from an early stage of the development on, and will hopefully increase the probability of a harmonized outcome. Yeah, I think probably in the interest of time, important here regarding the amount of data to be submitted, it's always important to strike a balance, not too little, not too much. You shouldn't be over-selective. It is always, we always recommend to proactively present potential pitfalls, and because in our experience, it's clearly a time-saving moment if this has been addressed proactively, and a CHMP recommendation has been provided how to address foreseeable problems. Importantly, try to be very scientific, try to provide comprehensive data from the literature. This will be helpful throughout the process. The question should be very clear, not too broad. Yeah, I think I can skip that. So probably to end this, I think there's a lot. We have seen a lot of advantages in an early on communication in order to streamline and to optimize protocols for the development of drug development tools. And of course, long term, this will speed up the time to regulatory acceptance of novel approaches and the time to new marketing authorization applications, and hopefully will save resources. I've included useful links to the EMA guidance, and with that, I thank you.
Many thanks, Thorsten, for this presentation. To reassure everyone, the presentations will be both downloadable and distributed to all the participants today. And the fact that Thorsten has skipped a few slides gives you an opportunity to go back to him and ask questions and see whether this direct contact works indeed as well as it was described. Now, without further ado, I would like to pass the floor to Amita Parikh from the FDA, who would speak about the drug development tool qualification at CDER at the FDA. Amita, the floor is yours.
Thanks, Magda. Can you transfer the controls to Tim, please? Tim on stereo, please.
Actually, it is put on your name, so.
Okay, that's fine.
Okay.
All right. Okay.
Amita's line has disconnected.
Can we transfer the speakers from Amita to Timothy? Could you help us with this, please? Amita? Okay. Do we have, maybe we should just continue, therefore, with presentations from the EMA on the qualification process, the formal qualification process from Maria Isaac. But maybe while we are waiting for colleagues to reconnect from the FDA, shall we check if there are any questions? So could I please ask the operator to remind people how to ask questions?
Thank you. If you wish to ask a question, please press *1 on your telephone and wait for your name to be announced. If you wish to cancel your request, please press the # key.
Hello.
Hello, Amita.
Hi, Magda. Sorry, somehow we got disconnected, but we logged back in.
Okay, so please do your presentation now, and we would collect the questions in the meantime. Please go ahead.
Yes, thank you. So thank you. Good afternoon. I'm Amita Parikh from CDER at US FDA, and I'll be giving an overview of the drug development tools, DDTs as we call them, that are currently in development at CDER. So DDTs are methods, materials, or measures that aid drug development. And during this webinar, I'll focus on the DDT qualification program that we currently have in place as one of the mechanisms to facilitate drug development. In the interest of time, I will not talk about other mechanisms and initiatives in development at CDER. So I'll cover the background, highlighting the events that led to the development of the DDT program. I'll outline the 3 pathways for qualification at CDER and provide contact details for further information. So the widening gap between scientific discoveries, the R&D investment, and the translation into medical products reaching the end user, the patient, is the impetus behind this rethinking of drug development enterprise that we call the Critical Path Initiative. And compounding this is the public expectation of safer, more effective, high quality, and less costly regulated products. We understand the need for efficiency in drug development and review processes, and the need for reduced uncertainty around marketed products. So one of the approaches to address these bottlenecks has been the development of tools such as biomarkers, methodologies, clinical trial designs, bioinformatics, etc., the much talked about efforts that hold a lot of promise. Now, these are monumental tasks and need collective efforts to forge forward. So the guiding principles such as collaborative efforts, toolkit development, sharing knowledge and resources, are the forces behind this critical path initiative for transforming discovery or proof of concept to a therapeutic product. FDA published the landmark paper on pipeline problems, the challenge and opportunity document in 2004. In 2006, FDA published the critical path opportunities list, and in 2011, the biomedical innovation document. Now, all of these outlined and articulated key concepts to foster enhanced medical product development. Several initiatives are underway at CDER to achieve these. As examples, guidances are in development. In addition, rare diseases activities, informatics programs, qualification of DDTs, etc., are also ongoing. I will provide an overview of the qualification program here in CDER, and Dr. Shashi Amoor will provide more specific details on one of these, the biomarker qualification pathway. But before I go there, it's important for you to understand how CDER is organized, because this will help orient you as to where various activities reside and who does what. So this is a chart showing the different offices in CDER. These boxes or offices each have specific responsibilities. They have review-related responsibilities and many critical path initiative-related responsibilities or tasks as well. The drug development evaluation tools we are talking about are coming out of different offices within CDER. Another important point to note is that even if a specific office is leading an initiative, the working group or the group working on any initiative may come from various offices or disciplines. So the working group can be multidisciplinary. Now, all of the activities that result in medical product development involve regulatory interactions. So this slide here illustrates the types of interactions that CDER staff is involved in. It's an oversimplified illustration, but gets the point across. So the first one on the left here is the regulatory review responsibility, which is the conventional review activities around the NDAs, the BLAs, including the pre-IND, the different phases of the IND stage, the NDA, the BLA review, and post-market activities. This is well-established, structured, defined, and well-trodden path of therapeutic review, and this, of course, is product-specific. But there's more to innovation than just NDA and BLA reviews. Development of guidances, for instance, or if drug developers have an important innovation or a concept that they want to discuss, that's not necessarily product-specific. So when external scientists want a non-regulatory, flexible, scientific exchange with FDA scientists, the voluntary exploratory data submission, the VXDS, the box in the middle here, is a novel pathway for this interaction. This involves sharing scientific information on issues of public health relevance that could develop into viable therapeutic products down the road. The other pathway of interaction is the DDT qualification pathway, which is also product independent. All three types of interactions really thrive and feed off of each other and facilitate drug development and innovation. So regarding this rightmost activity, the DDT qualification pathway, currently there are three pathways for DDT qualification. These are the clinical outcome assessment, the biomarkers, and the animal models under the animal rule. So what is this DDT qualification program? It was created by CDER as part of the FDA's Critical Path Initiative. It was created to provide a framework for development and regulatory acceptance of clinical outcome assessments, biomarkers, and animal models under the animal rule for use in drug development. It allows CDER to work with the submitters to guide them as they develop or refine a DDT for a specific context of use. CDER then rigorously evaluates the submission for use in the regulatory process, and a successful result is that of qualification for a specific context of use. And once qualified, drug developers will be able to use the DDT in the qualified context in settings such as INDs or NDAs or BLAs. And the benefits are that the CDER review group will not need to reconsider and reconfirm the suitability of the DDT if it is being used in the qualified setting. So this program has a shared vision and a mission. So to qualify and make DDTs publicly available for a specific context of use in drug development, and also to facilitate review of regulatory applications, provide a framework for scientific collaboration for DDT development, facilitate integration of qualified DDTs in regulatory review, encourage development of DDTs for contexts of use with unmet needs, encourage the formation of collaborative groups to undertake DDT development programs to increase the efficiency and lessen the individual resource burden that's incumbent with DDT development, and overall to encourage innovation in drug development. So now I'm not sure if you noticed that in addition to the term qualification, I used in association the term context of use a few times. It's very important to note that these two terms have a very precise definition from FDA CDER perspective. The reason is that a qualified DDT for a specific context of use carries a certain implication for regulatory decision-making in CDER. So what are the regulatory definitions of these terms? Qualification is the conclusion that within the stated context of use, the results of assessment with a DDT can be relied upon to have a specific interpretation and application in drug development and regulatory review. And context of use describes the way the DDT is to be used and the purpose of its use. A complete context of use should describe fully the circumstances under which the DDT is qualified and the boundaries within which the available data adequately support use of the DDT. And as I mentioned earlier, once qualified, drug developers will be able to use the DDT in the qualified context without the regulatory reviewers having to reconsider and reconfirm its suitability. And therein lies one of the benefits of DDT qualification. Now you may ask, can the DDT be used outside of the qualified context of use? Yes, but this involves case-by-case regulatory review to assess the suitability of its use in that setting, be it in an IND or an NDA or a BLA. And we have a draft guidance defining and explaining these terms and much more. In addition, we have a website on FDA's CDER webpage explaining a lot of the information that I have provided you thus far. These documents also discuss the steps for engagement. Dr. Amour will get into this in more detail. So the qualification program for drug development tools at CDER currently has pathways for 3 tools, as I mentioned earlier. These are the Clinical Outcome Assessment Qualification Program, Animal Model Qualification Program for models under the Animal Rule, and the Biomarker Qualification Program. And let me provide a brief description of what these are. The Clinical Outcome Assessment directly or indirectly measures how patients feel or function. It can be used to determine whether or not a drug provides a treatment benefit. COAs can also measure a safety benefit compared to other treatments. One of the distinguishing characteristics of COAs is who is doing the reporting of the outcome. Is it the patient or a clinician or another observer? Therefore, there are 3 components to COAs. Patient-reported outcome is a measurement based on a report that comes directly from a patient about his or her health condition. Clinician-reported outcome is an assessment that's determined by an observer with some recognized professional training that's relevant to the measurement being made and is based on clinical judgment. Observer-reported outcome is an assessment that's determined by an observer who does not necessarily have the professional training relevant to the measurement being made. Example, it could be a parent, a teacher, or a caregiver. So this is the COA. Now, what's the Animal Model Qualification Program? And I'm going to touch on a couple things here. At FDA, under certain circumstances, specifically when human effectiveness studies are impossible or unethical, the approval of drug or biologic products is permitted based on substantial evidence of effectiveness in animals. We call this the animal rule, and we also have a guidance on this. The selection of an appropriate animal model in which to establish efficacy is critical to the success of this program. An animal model is a specific combination of several things, namely an animal species, challenge agent, that's the material used to provoke the condition of concern, and the route of administration that produces a disease process or pathological condition that corresponds to the human condition of interest being studied under the animal rule. And there are two sequential parts to the qualification of animal models, the natural history and efficacy. The initial step is for the animal model to be qualified for the natural history of the disease or condition. And this means that the challenge agent induces a disease or a condition in animals that's analogous to human disease or condition. But remember that the main purpose of qualifying an animal model based on natural history is to use the model towards development and approval of therapeutic products. A qualified animal model is of greatest benefit when multiple drugs will be evaluated for efficacy in that model. So that's the animal models. And biomarkers, too, in the qualification program have a specific context of use, and Dr. Moore will provide more details. So just stepping back, the process of engagement with CDER is very similar for the three qualification pathways. It's comprised of three stages: initiation, consultation advice, and review. So with this overview of CDER's activities, specifically the DDT qualification program, the contact information for the three programs are provided on this slide. And if you recall from my earlier slide on CDER's offices, these activities reside in different offices. Biomarker qualification, for instance, falls within the Office of Translational Sciences, and Dr. Amor and I work in this office. This contact information is also on the CDER website for drug development tools. Thank you.
Many thanks, Amita, for this very clear presentation. Now I would like to open the floor for a couple of questions, if there are any. Could I please ask the operator to remind the procedure for asking questions?
Thank you. If you wish to ask a question, please press *1 on your telephone and wait for your name to be announced.
Please also remember that you can use the Q&A facility on your computer if you prefer to type the question rather than to ask it by telephone. Do we have any questions online?
There are no questions on the telephone.
I don't see any questions through the Q&A facility. Therefore, I suggest that we move on to the next presentation and the next part of our webinar, which would be dedicated to the formal qualification programs, both at EMA and the FDA. And I pass the floor to Maria Isaac, who is the scientific administrator at EMA and who would give us a presentation on the qualification process at EMA. Maria, the floor is yours, and Amita, could I please ask you to pass the ball to Maria on the Henry's name.
Magda, is Maria?
Under Henry, please.
Hi. Yeah.
Oh, under.
Can you see it?
No. Scroll back up.
Scroll back up. Magda, Natalie, Henry Fifth?
Henry.
Yes, right.
So give it to Henry. Okay, Henry.
Thank you very much. Maria, the floor is yours.
I am Maria Isaac, and I work at Scientific Advice. And what you're going to hear today is basically the view and the experience that we have so far in the qualifications opinions. But please keep in mind, like what Torsten has suggested, and this is a team effort, and a lot of people is involved here, and we have an excellent network of experts and advisors that really are people that are helping us. Then what you see here is thanks to the efforts of a lot of colleagues and networks. And for us, we thought that one of the suggestions was to put a clear example of how we think about it, And this is try to focus also in what happens when you have already done a lot of work and you think that you are ready for an opinion. For example, you want to have done a lot of work already in your patient-reported outcome or a methodology or model, or you have quite a bit of information about biomarkers. You already have a plan and you have developed a strategy. A lot of cases is the situation for IMI. I know, guys, that you have worked very hard and you have a lot of information there. And the question is how you're going to summarize all of these brilliant publications, all of this good work done, and trying to present it to the regulation. And to be sure that everything that you have produced can be useful and inform the regulatory pathway. Like I say, who you are is being presented by my colleague, and I wanted to give you a feeling of what things that we have been working already and what we have already collected, some information, some experience of what is going on. And for example, we have working in qualifications claims for diagnostics, prognostics of biomarkers. We're working in companion diagnostics in some developments. We have qualifications and have experience in trying to establish a standard of two, which is very important for you in biomarkers, trying to establish what will be a proper comparator in some studies, statistical plans. We do a lot of advice about qualifications about modeling at the moment and simulation, very exciting projects ongoing about this. And then definitely we will check. I know that some of you have wonderful preclinical matrix, platforms, and we are trying to review these for safety and for and trying to analyze. Then we have collected a bit of experience, and this is to give you the feeling of what we've been working so far. Like I say, we are a network. Something that is very important for us is patients, and you can see patients that get involved during these qualifications. I know that some people were thinking how they get involved or what kind of questions we ask them, and usually patients are very well informed and they have very good experts. Then most of the patients' representatives are people with a lot of knowledge about the diseases and a lot of knowledge about the implementation of these diseases, what it means for them. Then we found them very helpful and very important before giving an opinion, and they have also given the view about how this is going to be utilized. And usually it's also involved, is a lot of ethics involved in here, because some diseases, as you know, what we are working and trying to qualify are very early in the disease, and a lot of ethical issues about that, and we usually want to have feedback of them or how they will feel if this biomarker for early diagnosis can be in the market and what that means for them. And I think for us, the qualification opinion is very much the view or the regulatory view at that time of the CHMP of the acceptability or the specific use of the proposed method, such as a biomarker, in a research development program. And also, we would like to have a lot of data there, usually. But because some people say, well, what kind of data you use? We usually trying to have data that is already published, of course, but don't be surprised that for an opinion we will have to see some of your own data creating. And that has created a lot of questions about what happens if I am having a paper in Science, a paper in Nature, if you publish or we give it or we share with the public, if we put it in the website. This, of course, Science will be not be very interesting in the future to publish because it's already public knowledge. Then what this has created sometimes and has happened with some of our opinions is that what we have published there is the information that was made in the public domain, with the view that when you come in for marketing authorization and at the time of the marketing authorization, when this tool is being used, all of the data will be available for everybody to use. And I know that this is a big concern for some of you because what you are doing is great, very innovative, and I have a lot of questions from some of your groups when they've been discussing with about this. Then we understand how competitive it is, but the understanding is that by the time that you're going to have it in the website, it's going to be something that it will be also in the public side. And the context of use for us is very clear about how the proposed method and the research and development in the context is very specific to the indication, is very specific of what you want to claim. Are you going to use a biomarker for enrichment? Are you going to use a biomarker for disease modification? All of these details, they will be in the opinion, how you have collected this data. Then all of this information, it will be really, in one way, put in context your biomarker. Then for us, we understand, for example, if you don't have enough information about cutoff points, we understand that this will need to keep going and keep more information. We will be happy to give you a view about how far we can go with the current information that you have, as long as you will make the compromise to keep adding information in the future. Then we see it more as a work in progress together with you. Then any information that you are happy to share at that time, it will be added. And then the content will be defined. And every time that you bring more information, hopefully we'll be able to determine and inform better the context of use. Who can apply? As you know, consortiums, the networks. Our vision really is to help to speed up development innovation in Europe. And we really see how a lot of these good ideas that you have, we're trying to put in context with the regulators and see how we can help you in inform our guidance, our developments. Then that is the idea that we have. And of course, like I say, our recommendation, the final recommendation, we see it as a work in progress. The final recommendation definitely will be at the time of the EMA. When at that time we think that will be, the product will be finished. But we are happy to beginning to inform before that, because we know that guidelines and everything takes quite a bit of time to build up. The days, my colleague has explained you what we aim. For the qualification advice, this is aiming targets, but we're trying to do it, you know, qualification advice in 100 days. We're trying to aim for an opinion in 190 days. We definitely will give you something in writing in that guidelines, but it's true that it will be flexible to you. Suppose that you want to add more information. Not all the time this has happened, but you really, we have also qualification advices in 40 days. You know, some people really want an opinion quite quickly because it's very important for them, and we have done that. Then we are quite accommodating in how much discussion you want to have, and I think also what is important for you is that all of these qualifications, advices, and opinion, they are consulting and presented to the CHMP. Then for you is the possibility to present your ideas to the committee, the higher committee that we have, a scientific committee in Europe. Then I think it's a good setting for that, for trying to have this discussion on a higher level. Then I think it's the best place to present what you are doing. Yeah. This is to give you an idea of the numbers. As you can see, it was something that Peter van Bacchus is here. He was really the father of this with Hans Eisler. She, in 2007, with people like Marisa Papaluca, they were having with the FDA, they were having really the view of the future of what was needed for innovation, and this is the people that really created with the FDA. And the first ones in these qualification opinions, they were with the FDA and they were published. And they managed to create a procedure that we think that is quite flexible and open. And as you can see, in the last years, from doing 10 a year, in 2013, we have done already, we have 10 procedures by May. Then this is something that is growing and is growing very well, I think. And people is using it. And like I say, practical aspects, as you can see, is all of this information is what you know. You have the best teams in Europe working in this. You are IMI. And I think you know all of this information, and we will want to see, discuss with you, and trying to help you in any way to clarify what is the pathway better for what you are trying to solve and innovate. Keep in mind that a lot of times you want to claim something, and then during the procedure, the claiming changes. And the reason that usually change, and a lot of people ask these questions in qualification opinions, because you can see that. You can see what was your intention when you came. And then when you read our documents, you will realize how you started with this intention and then you finish with this claim. But basically it's trying to, a lot of times the regulatory language that we use, and in Europe, is to trying to have a common understanding of the different countries and different regulatory language. Then this is the process that what we give you at the end of the claim. Don't be surprised and it's different than what you wanted at the beginning. And it will depend a lot on the data that you have and how much we think that we can support the claim that you want. And keep in mind that there's a lot of guidelines there that can help you, the guidelines for evaluation diagnostic criteria, biomarkers, ICH. All of these documents are in the slides. It will be happy. It will be for you to give it a look. I'm sure that you have some feelings already about it. And like I say, it's a lot of things about how much some biomarkers are exploratory, how much they are used for confirmatory studies, definite, a lot of issues about, well, what happens, are I validated before phase 2, are I validated before phase 3. I can tell you one example, for example, if you are having a new indication that you want to claim with a patient-reported outcome and an indication that in Europe has been very challenging, I will advise you to try to validate before phase 3, because maybe the claim is something new, it's not in our guidance. Come early. This is something that is very well established and is in our guidance, but you want to give a new use, a new claim, I will be more relaxed, and then maybe you can come late. But this is something, I say, patient-reported outcome never been used in a clinical guideline. It's not in our guidelines. You want to use as a primary endpoint. You will need to validate that tool before phase 3. It will be too late for you, probably, because we don't have any experience. Again, for us, we understand what is the difference between standard truth. We understand what is the difference between a surrogate. We really understand, and you want to use very clear-cut populations, but sometimes also we will challenge you how this is going to be used in the market. We want you to think very quickly and very early how you're going to use this at the time of marketing authorization. We focus a lot about that, how it's going to be useful when you come into marketing authorization. Keeping in mind that we have even discussions with health technological assessment groups, and you can come for health technological assessment parallel advice about the utility of these biomarkers or the new tools, and what is the view of them, and what is the added value for them. And I think that is something also that you need to keep in mind in Europe, because at the moment is the way that all of this is going. Everybody knows about the new changes with NICE, how in UK this is changing very quickly, how NICE is going to do value pricing and all of this. And I think it's a good moment for you to begin to discuss with us, and we have a parallel advice experience here also. And he's the expert in that. He's the one that you can send your questions about that. The operator and monitor endpoints. This is to tell you about, very important for us, sensitivity and specificity. We really want to see what happened there. Don't forget about that. Is how we try to we look the data. And we are used to have retrospective, but definitely we need to have prospective data at the time of qualification opinion. And I say, this is the conclusions. We see it as something that is for you as a platform for communication with us, to trying to really put in regulatory wording all of the new innovation that you are doing and trying to inform the CHMP and trying to be sure that we are running at the same pace. And like Torsten explained very well, we want to see you sooner rather than later, but I understand that some of you have done a lot of work already, and we will be very happy also to see what you have done and trying to help you and trying to inform your decisions. And the outcome in the end, I mean, is that we know that what you want really is that these biomarkers, these new methods, they will finish in guidelines. And if we don't know what you are doing, it's going to be very difficult for us to inform our guidelines. Then sooner than you come into us, our guidelines, you know that they take a bit of time. Sooner than we can inform the guidelines, sooner than we can begin to have in our work program and trying to enforce the guidelines. And I think that is also something that you need to keep in mind in Europe. And Europe will work a lot in guidelines, and this is probably the way for you to begin to show what you are doing and trying to see what is a need for changing a guideline. If you are bringing new data, that will be helping us to also to plan the work program. And these are a couple of qualifications, and this is definitely things, like I say, a lot of people have helped me with these talk presentations, people here, Henrik, and all of them, that they've been doing all of the networking and information. Thank you very much.
Many thanks, Maria. Again, very comprehensive and a lot of hands-on and very practical information. Before I hand over to the next speaker, Shashi Amoor from the FDA, I would like just to ask you a question to which I would invite you to respond after Shashi's presentation, which is, how can I apply as a consortium or network for qualification if we are not a legal entity? So just please think about this question, and we will address it after a presentation by Shashi from the FDA. So could we please hand over to Shashi Amoor?
Magda, can you make it to Amita? Because
It's done, Amita. You can go.
All right, great. Thanks. So Shashi, you have...
Yeah, fine. Thank you. I'm Shashi Amoor, and I'd like to focus on one of the 3 drug development tool qualification programs we have at CDER FDA. That's biomarker qualification. I'm currently the scientific coordinator for the biomarker qualification program at CDER FDA. Here's a quick overview. I'd like to talk a little bit about the biomarkers in drug development, and next about the CDER's biomarker qualification program, and end with a few take-home points. Biomarkers can become integrated in the drug development process through the drug review submissions on a case-by-case basis at CDER. However, I would like to tell you first that these can help in understanding pathways underpinning disease, in understanding the mechanism of action of therapeutics, and may be used in multiple ways in clinical trials. Biomarkers can be used for multiple purposes, as I mentioned. They could be stratification or patient selection. They could be for monitoring response, and in a few cases, they may serve as surrogates to the clinical endpoint. Biomarker tests may be needed as companion diagnostic in identifying the right patient subpopulation for prescribing the therapeutic for safe and efficacious use. So these biomarkers can become accepted or integrated in the drug development process through the drug review submissions on a case-by-case basis at CDER FDA. Biomarker data can be part of the exploratory studies or can play a part in clinical trial design. Biomarker tests may need to be developed along with the therapeutic, and they become companion diagnostics to the therapeutic. The advantage of this pathway is that the biomarker becomes accepted in the context of the drug through the drug review process. The limitations of this process are that, 1, the biomarker will be useful only in the context of the approved drug most cases, And 2, the use of the biomarker needs to be discussed with the review division for every new regulatory submission. Exploratory data, as you know, is available for hundreds of biomarkers, if not thousands. The gap between these biomarkers and application to drug development needs to be bridged. Acceptance of biomarkers through the IND, NDA, BLA process is one way that's shown as a small tile that a gentleman is holding up on the right side of the screen. A more robust bridge is biomarker qualification, since biomarker qualification is meant to be a process that helps multiple drug development programs. I'd like to give you a brief history of the biomarker qualification program at CDER FDA. A pilot biomarker qualification process was initiated in 2007 with the PSTC submission on nephrotoxicity biomarkers in a joint effort with EMA. Using this experience gained through the pilot process, the biomarker qualification program was introduced in CDER in 2009, and as Dr. Parekh mentioned earlier, a draft guidance on qualification process for drug development tools was issued in 2010. This guidance is currently being finalized. Biomarker qualification, again, repeating what Dr. Parekh had said, can be defined as a conclusion that within the stated context of use, the results of assessment with the biomarker can be relied upon to have a specific interpretation and application in drug development and regulatory review. The website and the email of the point of contact for the biomarker qualification program is provided on this slide. I'd like to provide some details on the context of use. Context of use is a comprehensive statement that fully and clearly describes the manner and purpose of use for the biomarker. The context of use statement would describe all important criteria regarding the circumstances under which the biomarker is qualified. In addition, the qualified context of use would define the boundaries within which the available data adequately justify use of the biomarker. This is an important consideration since the context of use determines the kind of data needed to support the use of the biomarker in drug development programs upon qualification of the biomarker. This slide shows some information on biomarker qualification program at FDA that answer many frequently asked questions. One question that's often asked is, who can submit a biomarker qualification application to FDA? Submitter can be a person, a group, organization, or a consortium that initiates a biomarker qualification proposal using the procedures described in the guidance. At CDER FDA, no fees are charged for submissions to the biomarker qualification program. And I think I'm repeating what Dr. Parekh said. Once qualified for a specific context of use, a biomarker can be used by drug developers for other applications without re-review. Oftentimes, the submitter may choose to aim for the low-hanging fruit or an easier-to-reach context of use. However, incremental expansion of the qualified context of use over time may be undertaken. Biomarkers considered for qualification at CDER FDA are conceptually independent of the specific test performing the measurement. We do believe that the analytical validity of the test is very important so that we can trust the results. Thus, however, the biomarker, if it's qualified by CDER, the test used to measure the biomarker is not automatically cleared. CDRH or CBER are the centers responsible for clearing biomarker tests. Finally, biomarker qualification is a tool for drug development and not for diagnostics or for companion diagnostics. Again, CDRH and CDER or CDER are the centers responsible for clearing tests or devices for diagnostics or companion diagnostics purposes. So far, 3 biomarker qualification submissions have successfully gone through the biomarker qualification process at CDER FDA, and these FDA-qualified biomarkers are listed on the drug development tool webpage, and it's shown on this slide too. So basically, we provide information on the name of the biomarker, the submitter of the qualification submission, of course, and the date the biomarkers were qualified. We also provide some links to supporting information. There are some important considerations that are needed prior to biomarker qualification effort. Some preliminary data that's promising and reproducible, a possibility that this biomarker may be useful in drug development, an available robust assay, preferably analytically validated for the biomarker to increase confidence in the data generated or to be generated. It's also important to put some thought into the way or ways the biomarker can be used in drug development of multiple programs. Context of use or contexts of use, which could be more than one, what is known already, what are the gaps in knowledge, what more needs to be done to qualify the biomarker. These are important considerations. I'd like you to remember the context of use is not written in stone and it evolves as the biomarker development progresses. However, it's good to consider possible uses of the biomarker in drug development early in biomarker development. The biomarker qualification process can be divided into 3 stages, as Dr. Parekh has explained: initiation, consultation and advice, and review stages. I would like to provide some details on these stages. This slide shows you details of the steps involved in the initiation stage. After receiving the initiation request from a submitter, generally through an email sent to the biomarker qualification program, the program communicates with the submitter, which often include a telecon and a follow-up email with instructions on how to submit the letter of intent or LOI. If not suitable for submission to the program, we direct the submitter to other paths, such as CDRH for device or test clearance for clinical practice diagnostic tests, or to the review division if the qualification is in the context of a single drug under IND, NDA, or BLA review. After receiving the LOI, if the information is deemed adequate by the biomarker qualification program, then requests for reviewers are sent to the office directors and the office liaisons, and the biomarker qualification review team, or BQRT, is formed. This will represent expertise from different offices that would be pertinent for that submission. The BQRT reviews the LOI and provides specifications for the initial briefing document to be submitted as the next steps. These include advice, suggestions, and comments. These are sent to submitter upon clearance. Submission of the initial briefing document by the submitter marks the beginning of the consultation and advice stage. BQRT reviews the submission and provides comments to the submitter prior to the face-to-face meeting. The submitter then usually gives a slide presentation at the face-to-face meeting and selects topics or issues for discussion with FDA. Meeting minutes are generated to capture the discussion and action items and sent to the submitter. This is often an iterative process until the submitter and FDA determine that the biomarker development is complete. Additional briefing documents may be submitted as needed, and face-to-face meetings or telecons may be held again as needed. The next stage starts with the submission of the full qualification package. An internal BQRT meeting is held to discuss review expectations, and a date for the internal mid-review is determined. At the mid-review meeting, the BQRT discusses the submission and determines the need, if any, for additional information or for the need for modifying the context of use. The BQRT also discusses the need for wider presentations, such as regulatory briefings. If it's determined that wider presentations are needed, these are then organized. If additional information is needed, request is sent to the submitter and the material reviewed upon receipt. Once all the discipline-specific QRT reviews are complete and with qualification recommendations, the program then determines whether a consensus on the qualification of the biomarker or biomarkers has been reached. If yes, a qualification clearance package is then prepared. The biomarker qualification program sends a letter to the submitters informing them of the CDER's qualification recommendations, and these recommendations are also posted on the Drug Development Tool public web page, in addition to redacted qualification reviews and the executive summary. In addition, the recommendations are posted as a draft guidance appendix to the DDT qualification guidance on the FDA public page. I'd like to give you a brief update on the biomarker qualification submissions we have currently. As I mentioned earlier, 3 submissions have been qualified so far, and 1 is in the review stage, which I didn't mention earlier. 13 submissions are in the consultation and advice stage, and 6 are in the initiation stage. As you can see from the figure on the right, in the consultation and advice stage, roughly 70% of the submissions are for clinical biomarkers and 30% for non-clinical biomarkers at this time. The early submissions were mostly for qualification of non-clinical biomarkers, and we have received a number of submissions for the qualification of clinical biomarkers in the last 2 years. The types of biomarkers that we see in the biomarker qualification submissions are preclinical safety biomarkers, clinical safety biomarkers, efficacy response biomarkers, and patient selection biomarkers. As you can see, roughly 50% of them are for safety-related biomarkers. Consortia appear to be an efficient path for biomarker qualification. This may be because of the sharing of resources and effort needed for qualification by multiple parties. Some examples are shown on this slide. Here are the take-home points that I have. Biomarker qualification is not a requirement. The case-by-case approach remains a valuable process. Currently well-established, widely used biomarkers do not require formal qualification. Again, biomarker qualification is intended for biomarkers that will be used in multiple drug development programs, and in our experience, consortia or collaborative groups are likely to be the sources of biomarkers for qualification. Lastly, biomarker qualification is a voluntary process, and no definitive time clocks are associated with it. I'd like to thank Dr. Woodcock, Dr. Ruffman-Garner, Dr. Walton, Dr. Zinne, and Marianne Noon. And again, the point of contact person for any questions related to the biomarker qualification program or process is Marianne, and her email is listed here. And please feel free to send me any emails with questions also. And I'm done. Thank you.
Many thanks, Shashi. Before we go further, I would like the operator to remind us about the procedure for asking questions by telephone.
Once again, as a reminder, if you would like to ask a question, please press *1.
Thank you very much. And also, please remember to use either the Q&A or the chat facility in order to ask your question. And here comes the 2 first questions. So I'd like to remind you the one which was asked just before Shashi's presentation. How can I apply as a consortium or network for qualification? EMI consortia are not legal entities. So without being a legal entity, can I apply first to the EMA and then to the FDA? Who would like to respond? Yeah.
Hi, this is Spiros Vamvakas in the EMA. You do not have to be a legal entity to apply for a biomarker for the EMA. It can be the consortium. It can be one company of the consortium. It can be the SME of the consortium. It is up to you. The important thing is that the biomarker is developed in the context of use for regulatory purposes for development of drugs. That's a short answer.
Thank you very much. On the FDA side?
I mentioned in one of my slides that anybody can be a submitter. It could be an individual, it could be an organization, it could be a collaborative network or a consortium.
Thank you very much. I hope this clarifies. Another question that we have is, could you comment on the opportunity or chances to bring forward for qualification and advice a set of biomarkers rather than a single biomarker for a certain use? So I don't know who would like to reply first, maybe from EMEA?
Yes. It depends upon the biomarkers. For example, if it is several biomarkers in terms of several proteins which would be predicting for toxicity, which could be answered by the same qualification team with the same expertise, yes, it could be several biomarkers in one application, and it rather makes also sense. Now, at the other end, if you would like to qualify a clinical surrogate endpoint and a biochemical biomarker in the cerebrospinal fluid, this cannot be one because this requires two sets of experts. So it's the nature of the biomarker and the required expertise which makes us putting together in one application certain biomarkers or splitting them up. And we had this discussion, and we have the opinions actually in our website on the biomarkers in Alzheimer's, how they were split based on the nature of the biomarkers.
Thank you very much. How about the FDA?
We have examples for all of the above. We have seen single biomarkers. We have seen composite biomarkers where it could be a panel of biomarkers used for a single context of use. Sometimes we have submissions where there are multiple biomarkers and maybe two of the biomarkers are good for one context of use and three others are good for another context of use. So all the possibilities are good.
Sounds very good. Do we have any question online?
Your first question comes from Asaya Cloud. Please ask your question.
Actually, this is David Laurie sitting with Saya Cloud. I have a question for both EMA and FDA. I just wanted to ask what is being done, if anything, to try to harmonize the document requirements, the briefing book requirements, for example, and also the timelines for review, particularly with respect to parallel advice.
Who would like to address this question on the EMA or on the FDA side? Shall we start with EMA?
On the EMA side, we do not have really a template which has to be followed for the biomarker submissions. We have to have, it's as simple as that, the questions, the company's positions, you have to have a background and attachment. So I'm not sure if an alignment is necessary. We do not really follow something like a CTD structure or an EMA-specific template. So we could be flexible as long as the scientific information is available in the application. Regarding the timelines, it is actually, again, depending very much upon the extent of the data, the scope of the biomarker, and also the applicant's needs to receive an answer. The biomarker numbers are still manageable. As Maria showed, there are 10 to 20 perhaps this year. They are not in the 400s like the regular scientific advices where we are forced to have very, very strict timelines. So we can be more flexible to accommodate the applicant's needs.
Thank you. An opinion from the FDA side?
We have 3 examples in the recent times where the submitter has sent the submission, the LOI or the letter of intent, to both EMA and FDA. From an FDA perspective, we are fine with the template used by EMA because what we care for is the content, not so much for the format of the letter of intent. So we have accepted the same information in that template. And in addition, we wanted a couple of things, and those are listed on our website, the cover letter, etc. And so that was easy. The first step is the easy part. And after that, we do not have any time clocks currently associated with the biomarker qualification process. But in terms of time for the initiation, maybe it takes about a month or so. And then for the consultation and advice, it varies. It's anything from 2 to 4 months, depending on how ready the submitter is and if we need any additional information or clarification at that time, and so on. So, but we are working with EMA on these 3 submissions, and we'll be exchanging our comments and suggestions, and also we participate as observers in their meetings, and they will be participating in our meetings as observers. I hope that helps.
Many thanks. Does that reply to your question?
Partly. There is the aspect of the feedback from EMA at its pre-submission meeting, and then having separate feedback from the FDA's BQRT, which comes. Well, we're experiencing that coming a few weeks later, and therefore we're having to prepare a final briefing book for EMA, followed by an adaptive briefing book for FDA. I'm just wondering, particularly with concern for that point, whether that is an opportunity for harmonization and a consolidated feedback from in a pre-submission context from both EMA and FDA, so that we only work with one data package.
We may not have a response to this question today. I would still leave the opportunity to EMA or FDA to comment, but maybe we should just leave them time to discuss it and to come back later if appropriate. But are there any comments to this question, to this particular point?
Hi, this is Shashi. Yeah, what I can say is that if the submitter is willing to wait till both sides are ready with their comments, this can happen, but oftentimes, you know, the submitter may want to go ahead with one of the 2, that's EMA or FDA, and not wait. So if it's possible to wait, we could try to harmonize.
Okay, thank you very much.
Just Thorsten Vetter from EMA. Regarding the 3 procedures that were just referred to, I think that highlights if you are planning to come for a parallel FDA-EMA, it's very worthwhile to plan that sufficiently early and to come to both agencies and ask dedicatedly for timelines. I think that helps the process.
Thank you very much. Do we have any other question online?
There appears to be no further questions.
Okay. I just have one. What is the rate of failure in these procedures? How often does it happen? What is the percentage? Just roughly, because that may also inform how serious the procedure is. So how often does it happen?
From the EMA point of view, we do not understand the question regarding failure. There is no failure in this procedure. As Maria mentioned, you may submit for a qualification opinion based on the fact that it is your view that the development has been finalized and the data are fit to qualify the biomarker for a context of use. If the EMA expert, the regulatory expert, concludes that some additional data are needed, then the applicant receives back, instead of a qualification opinion, a qualification advice, or a more limited qualification opinion, depending upon what is missing. But I wouldn't say that this is a failure. This is an ongoing dialogue in the course of a scientific development. It is not a marketing authorization for a new product. This is a completely different concept.
Thank you very much for this comment, because it is extremely important to understand what is the nature of this, and that it can only enrich the the scientific work that is being done. I suggest that since we don't have any further questions, we move to the last presentation of today by Nathalie Seigneuret, who will try to summarize, and not to compare, but to summarize and to put side by side the two processes in EMA and FDA. And may I just ask Amita to move the ball to Nathalie?
Yes.
Thank you.
Thank you very much, Magda. And before going into this summary on the similarity and differences, I would like to make just a couple of points which hopefully will help you as a consortium. First of all, some of you have already started some interaction, but most of you may not have been familiar with this process. So we hope first that this presentation have shown you first the value of engaging with the regulators in order to help you to progress with the project, and ultimately, as it was mentioned several times by both EMA and FDA, is to have a concrete final outcome and hopefully to have some impact on the guidance or to update the guidance, so to really be a driver as well in the scientific work, but also to show you how approachable the regulators are and that their commitment in their role in supporting innovation. And saying that, I think it's important for you to realize that with this different procedure and timelines, it's important for you to go back to your project and to think, for those who have projects with output of regulatory relevance, where or at which time it would be best to start the discussion. And if I go to my next slide with the first table summarizing, oops, I cannot go to my last. Oops, sorry. Okay, it's a small table, but hopefully you will be able to read it once you have the slide. But as it was pointed out several times, come as early as possible, because that will be a way of helping to shape not only the process and even to give you some output on your project and to help to progress, and to plan as well when you will come with the results for the qualification. And as it was mentioned, this is not a fail or go, no-go decision. And the fact that having maybe a defined biomarkers or tool at the end within a specific context of use may help as well to understand which other project or which other research will be needed if we want to expand the research of use within this project or other projects. So this is important to keep in mind. And going back as well to the communication and dissemination, I would just make one point, is the fact that this is completely in line as well with the concept of IMI, of sharing and communicating the results. So the publication of actually the outcome is very important there. So saying that, I think what we have heard, and I won't discuss again in details what has been presented, but in fact is to show that there are more similarities than differences between the two approaches, and that the message here is really that consider to come as early as possible, to start the initiation pre-submission, to have consultation and advice, and then to have the review itself. The scope is more or less slightly different, but as we have seen in the context of the FDA, there are other programs as well where other activities can be discussed for some of the other tools. The definition, again, of qualification is the same, and what is important is the context of use, which define really where the tool can be used and the boundaries for its use. Who can apply, whether it's called an applicant or submitter, again, that can be anybody. So as a consortium, you can apply. Maybe just one point to keep in mind for the EMA level, it may be worth to consider if you have an SME as part of the consortium, maybe to go through the SME for the fee reduction, but there might be other way, and that definitely will deserve to be discussed at the pre-submission with the actual team to see what would be the best for the application. If I move to when to submit, again, what was clearly put from both sides is to do it as soon as possible in order to have a prospective advice on a prospective dialogue rather than a retrospective or being at the late stage of the project where actually everything has been already done. Now, how to submit? Here, from all the slides, you have seen the different contact points and the fact that, in fact, the same letter, in a way, can be submitted. What is important to remember here, not so much the format, it's much more the data to be submitted. And when we talk about the documentation, what was important to flag is that it's to make the balance between not too much information, but not too little information. The regulators need to have sufficient information in order to understand as well the project, what you are doing, and what is the tool which has been developed and what will be the context of use. The output of the both procedure would be really on both sides official guidance, and that will explain the way the method has been qualified for the specific use and the specific context of use, which may be then used as part of an application for marketing authorization. Confidentiality, whatever will be before the actual qualification recommendation will be confidential, and once there is the qualification opinion, then that's where it will go to the public domain. The length of procedure, my understanding is that even if there are timelines, it can be flexible, and on the EMA and the FDA, less defined timelines. But again, because it has to be seen as a much more dialogue rather than a rigid procedure, this may depend as well of the complexity of the process, but as well of the amount of data provided. So I think here it's important to remember that to think about that when you try to find in your actual project where it will be the best timing to start thinking of engaging this dialogue as early as possible, as we said, but as well in terms of your duration of the project, which will be the relevant milestone in order to have this interaction in place. In terms of the follow-up, again, the advice or the qualification opinion is not set in stone. If there are further data coming in which may expand the context of use, or if which, in terms of the advice, if further data will come which may give some new information on how to progress the development of the tool, this again can be again discussed in order to maybe reshape the process or the project as it is defined. And I think this is my last slide, and again I think that this presentation has gave all the presentation I gave you the flavor that the importance of thinking on not forgetting the regulators in those projects where you're developing tools which will be of regulatory relevance. And I think I will stop here. So Magda, I will pass you the floor again.
Thank you very much, Natalie, for this very useful overview. I think it is the first time I saw these two procedures compared side by side, which is great. I would just like to check with colleagues from EMA and FDA whether they had anything to add to that, or if they would like to point at, something very specific as a last comment before we close this session.
No comments from the EMA. Thank you very much to everybody and to Natalie for the excellent comparative table.
On the FDA side?
Contact us early and we'll be very happy to help. That's all I have. Thank you.
Many thanks. I would like really to thank everyone who contributed to this session today. Through presentations, a lot of preparatory work, Natalie for providing this comparison of the two procedures, Torsten and Maria for the presentations, Amita and Shashi as well. I would like to thank all the participants who are listening to us, to remind you that this webinar is recorded and a link would be provided, that the presentations will also be distributed and available online, so you would be able to look at them again. And again, don't hesitate to ask for more questions at EMA and FDA and contact the contact persons indicated on the slides. Thank you very much, everyone, again, for your attention.
Magda?
Yes.
Magda, sorry, if I just may, I think it would be nice for us and to have the feedback from those of you who have listened as well, how useful you have found this information session. And again, if you have some questions, of course, please contact EMA and FDA directly, and as well within the office and EFPIA office as well. We are here to help, and therefore, if we can be kept in the loop, or at least if we can help as well, please do not hesitate.
Thank you very much, Natalie. And I think that we would certainly come back to you. Thank you also in advance for your feedback. Together with the presentations, you would receive a short feedback form. Please take the time to respond to it and let us know about any questions that wasn't addressed today and that you would like to see addressed in the future in any training or information session. Thanks to everyone, and see you soon. Thank you.
Thank you.
Thank you. That does conclude your conference for today. Thank you for participating. You may all disconnect.